This is Voices in Validation, brought to you by IVT Network. IVT Network is your expert source for life science regulatory knowledge. Voices in Validation brings you the best in validation and compliance topics. We interview industry experts from pharma, biotech, med devices, and laboratories. Here is the host of Voices in Validation, Stacy.
Thank you for joining us once again on Voices in Validation. We're glad you're back with us. And this week we are going to be digging in a little deeper on some cleaning validation. Visual inspection of equipment has always been an important element of the cleaning validation program, and establishing visible residue limits, or VRL, which we'll refer to it today, is extremely valuable in setting a baseline for cleanliness, highlighting viewing conditions, and defining a methodology for performing routine checks on cleaning validation procedures. Understanding the results of testing and reproducibility of test results under varying conditions allows for consistency across teams and lessens risk of error in cleaning validation activities. Here to speak with us about that today is Rich Forsyth. Welcome back, Rich. Great to have you with us again.
Thank you, Stacy. It's great to be here.
Absolutely. So, Rich, let's just start off with the basics first. Can you kick us off by providing an overview of the visible residue limits program parameters and how it should be incorporated in the overall cleaning validation program? And then
Sure.
What does ruggedness refer to in this case? So start with the first part, please.
Okay. Visible residue limits are, the way I define them is the amount of residue that all observers can see on a given piece of equipment, or use coupons, stainless steel, stuff like that. The way you determine what parameters to use, you should go out to your facility and look at the areas where your personnel will be doing visual inspections of clean equipment. Then you could measure the level of light intensity out there, and it'll vary from room to room. So you want to get a range. Look at your equipment. Typically, you'll get as close as you can to look at the clean surfaces. So, and that'll vary, because if you have pieces that have been taken apart, you can get as close as you want. So typically, a foot to 2 feet. But for larger pieces of equipment, you might not be able to get any closer than 10 feet to some surfaces. So you want to do a distance parameter when you do your VRLs. And then the residues themselves, a lot of them will look similar, but some will look different. So it's good to, I recommend that my clients do VRLs for all of their residues, so they get a survey of everything that they'll be looking at.
Sure.
The most critical parameter, though, is the viewing angle, and that's just what's the angle between your eye and the flat surface that you're looking at, because that can change dramatically what you're able to see.
Oh, right. I would bet. And so the second part of that question is, because we hear the term ruggedness being used with visible residue limits frequently. So what does ruggedness refer to in this case?
You would think that the people's ability to see residue is going to vary from person to person, and it will to a small degree. So when I have my clients do VRLs, I recommend that they get as many people involved as possible. To do the actual observations, you can do that pretty quickly. So the more people you can get involved, and then the better. And then the lowest level that everybody can see, or just about everybody, there could be one outlier. That's what you define as your visible residue limit. There is, as I tell people going into this, it's like there's no right or wrong answer. Mm-hmm. It's what you can see.
Right.
And doing that, it generally the variety, the difference in the levels that people can see doesn't really vary that much. So I get a number of people in, get as many different residues as I can, and have them come in and do the observations. Logistically, it's tough if you have a large facility with a lot of residues and a lot of people, it's hard to make all those combinations fit. So what I'll tell them to do is we'll get a representative number of products. Typically start with your hardest to clean products and do the visual limits on them and get everybody involved, the people who inspect the equipment, the people who actually clean the equipment, the supervisors get in between there. So have them all work at establishing the visible residue limit. That way you have a lot of redundancy built into the system, and that works into the ruggedness of the VRL.
Great, thank you. So how does VRL differ or compare to ARL or acceptable residue limits? And are we ever in a position where we have to choose one over the other?
Yes, if for very potent products like a steroid or a hormone or cancer drugs, your acceptable residue limit, and that's the amount that can carry over without threat to patient safety, will be fairly low. And for things that are really potent, it actually could be lower than the visual limit. However, for most products, the visual limit will be lower than the acceptable residue limit. And then it's just a matter of how much lower is the visible limit. For safe products, the visible limit's anywhere from 10 to 100 times lower than the acceptable residue limit. So,
is that because we just accept that there may be some residual product in whatever other products they're making, or why is that? Why would the visual limits be lower than the acceptable limits?
Because the amount you can safely ingest for something that's fairly safe actually gets pretty high. In that case, the equipment would be visibly dirty and the patient could still be safe. Now, that being said, the first criteria for clean equipment is it has to be visibly clean. So that's kind of like your built-in safety margin. If your visual limit is significantly lower than your patient's safety limit or ARL, then you can pretty much say if the equipment is visually clean, then my patients, there's no risk for my patients or for the patients, of the next drug.
Sure, that makes sense. So what parameters matter most then in a VRL program?
Oh, okay. And in order of importance, as I already mentioned, the angle is the most critical. And I've done a lot of work in different angles, and then typically we would do that. And generally speaking, your visual angle should be greater than 30 degrees from, and 30 degrees, if you think about it, if your surface is laying flat on a table, 30 degrees up to your eye. So that type of viewing angle. Greater than that is okay. If you think about it, if you're looking straight at a surface and there's a light in the background over your head, then you could get glare from the light. So you have to be careful about that. But overall, by far the angle is the most critical. Distance has some bearing on it for sure. Most of my data shows as long as you're less than 10 feet away from the surface, then you should be able to see the visual limit. So that's a pretty wide range of distance, and you can cover just about all of your equipment except for very large pieces.
Okay. Yeah. I mean, 10 feet seems like a pretty fair distance. I mean, that's kind of far away. Although I guess depending on what part of the equipment you're looking at or what exactly you're looking for, sometimes you need that distance to be able to see.
Right. You're prevented from getting any closer on some pieces, like a large fluid bed dryer. The columns go up pretty high. Tanks are a great example. Any large tank, you don't want to break the barrier, looking into the tank very much. I was at one place where they had a camera and they had on a cable and they could drop it into tanks, so that kind of eliminated that. And that's an interesting application if you have a lot of tanks, then you could eliminate that distance problem. As far as the light level goes, did a variety of light levels. It generally engineering criteria for manufacturing areas define a light level as around 700 lux, and that's fairly bright. So I've done all the way up to 1400 lux. Looked at using a handheld spotlight, because people will do that to look at tanks, and then taking that all the way down to 100 lux. And the data is pretty consistent regardless of the light level. So kind of fell in that as long as your light level is greater than or equal to 200 lux, you can still see your visual limit. And that gives you a fairly large comfort level as far as the light level goes, and the distance. So the most critical one there is the angle.
Excellent.
That being said, the residues themselves are a little bit of importance as far as being able to see them. Salts will generally dry to a white residue. Sometimes you'll see a gray residue, and then a lot of bioforms will actually go dry clear, and then you have to be a little bit concerned about that. But that's where you get a feel for that when you actually establish your visual limits, and that's why it's good to do the entire range of your products, so you get a feel for what your residues are going to look like, and you can train your people to understand that.
Right. That was the comment I was going to make. It seems like you have to really know what products, what to expect from the products you're using. And then, depending on which you're using, like, no, this is going to dry clear. So we need to look for what? We need to look for something that looks wet, something that looks shiny, something that looks
exactly,
like muted or matte finish or what? I mean, like, you've just figured out, right?
Yes. And that's critical for the training for this.
Right. So that's what I mean. Yeah, it would be super important that everybody who's working in that area knows the nuances of the products and knows what they're looking for, so that they can maximize their cleaning abilities.
Correct.
Rich, what are the regulatory expectations around visually clean examinations? And can you talk about observations or warning letters that you've seen related to this topic?
Yeah, actually, I did my first work on VRLs probably 10 to 15 years ago. And initially there was some excitement about it, and then it just kind of settled down. But recently, one of the hot button items for the regulatory inspectors is that people who do their visual inspections of their clean equipment be qualified. And how do you qualify somebody to do a visual inspection? So there's been kind of like a resurgence in interest in VRLs, because you can actually quantitate what people can see by establishing a VRL for your products. Mm-hmm. So it kind of plays into or supports the ability to qualify people to do visual inspections. And you have data to show that they can see this and see it consistently.
Is this a formal qualification where you're getting some sort of certificate after testing, or you know what I mean? Like when you say certifying or verifying, like I'm just trying to picture how formal this process is.
You do it in-house typically.
Okay.
For years, they've qualified people who work in laboratories by giving them test samples and have them analyze them, and they have to get close to what the established number is. So it's kind of like that. It's you do your visual limits, your VRL, and everybody who is involved in establishing that, okay, now they can see the residues. Then the other parts are training the people to how to look at equipment. So that would be like field training in the facility.
Right.
And in the past, they didn't do that. It was go look at the equipment, tell me if it's clean. And that's so understandably, the agencies are a little concerned about that because the visual inspections, in my experience in the industry, have been somewhat inconsistent. It's been very qualitative. How's that?
Right. That makes sense. I mean, you want, the individuals who are doing the inspection to be familiar with it, to have been trained on what to look for, and to not just be someone like, hey, Joe from this department, go check the equipment, right? So it makes sense that there's a little bit more formality around it. Like you have specific people who are doing this job within your department so that you can have consistency and accuracy, I would think. About observations and warning letters, have you seen any related to either not having someone who's been qualified within the agency, or that they've just been too lax with the way that they're handling this?
Companies have not been qualifying people to do visual inspections. So now the agency is telling them they have to do it.
Okay.
So that's where it's been a hot button citation probably for the last 2 years or so.
Okay. So as of 2 years ago is when they really wanted people to start taking this a little more seriously and having a qualified individual, would you say?
Yes. Yeah, I would say it's been about that long.
Okay. And what do those warning letters or observations look like?
It's just an observation or a citation that the people who do the visual inspection of your equipment are not qualified. I mean, they don't tell you how to do it, just like everything else.
Right. I didn't know if they were connecting it to other observations like failures in the product, failures in the cleaning process, or something like that. It's
if they can tie to a specific observation they've seen, then that makes their jobs a lot easier to cite them for it. But I've been places where they've been cited for it just because they don't have a program.
Well, we do know that the agencies care about our SOPs and making sure that we're following our own protocols and that we know what they are. So that makes sense. Rich, can you speak about the methods of data analysis in VRL activities?
I can. I mean, I'm not a statistician myself,
right.
But I've done some rudimentary statistical analysis of groups of visual limits. I've done visual limits of the active ingredients, excipients, and individual excipients and formulations. And the reason I did that, initially when I was developing this, I was in a R&D facility. We were doing clinical supplies. So the formulations would change. So we didn't have a lot of final formulations to work with. So I did the APIs and the excipients individually. And then when I could get a formulation, I actually compared the formulation to each of the individual components of the formulation. And that was fairly interesting. Sometimes it matched up, sometimes it didn't, but but I generated a lot of data, and the statistically, the groups were not distinguishable, meaning the visual limit for an API was similar to the visual limit of a formulation or the visual limit of an excipient. And I could conclude from that, okay, well, if you do the visual limit of the API, you're probably good. And that works for a development facility. If you're in a commercial facility, it only makes sense to do the final formulation because that's what you're going to work with time after time. I have seen other statistical treatment of VRLs. They're somewhat interesting, but they're more to me, they're more concerned with the statistics than with the visual limits and how they're used.
Understood. Yeah. As data analysis sometimes can be.
Yes.
So describe for us then the use of random and sequential presentation process and the impact on observers conclusions then?
Okay. Yeah. I mean, when I started, I always presented my residues in sequential order, meaning from highest level of residue down to the lowest level, and also included a solvent blank in there. And I told people, it's when they were looking at these, again, there's no right or wrong answer. So just tell me what you can see and what you can't see. But over time, the concern was, well, since they're going down in amount, are people being kind of prompted to be able to see the lower levels?
Right. You automatically mentally assume this is going to be less than the last because we're moving on down the list, right?
Right. And a lot of times we were able to actually see the lowest residue that was spotted. So it kind of supported that, although we would have residues once in a while that you couldn't see at the lowest concentration. So it's like, okay, then I'm not too worried about the sequential presentation. But somebody brought it up, so it's like, oh, that sounds like an interesting experiment. So I got, I think it was like 5 residues, and brought in and did a random presentation, and brought in 10 people to look at them, and they identified which ones they could see and which ones they couldn't, and they didn't know which was which. And then I took 2 of the residues and put them in sequential order, and brought back 2 of the people who I used. For the random, I think had 10 people, and I just brought back 2 of them for the sequential observation, and their results were exactly the same. So I published that study back in 2009, basically concluding that there really wasn't a difference in presentation. Now, I'll qualify that by saying, if the people were trained, what they're looking for, they're used to running these type of studies. It's not real complicated, but the first time you bring somebody in, you have to explain to them what's going on and what you're looking for.
Sure,
so
that makes sense. Yeah.
And typically I'll still run any of these studies sequentially, because frankly, it's easy. The bookkeeping is easier for it.
Listen, sometimes that is part of it, right? Just having your own methods of organization and something that makes sense to the team so that everybody can be consistent with what they're doing.
Yeah. It's a random presentation is perfectly acceptable. You just have to make sure you keep, very good track of the samples and which is which. Right.
Does it matter amongst teams if they're doing it like 2 different ways, or should a team be very consistent? Like if as a team they're going to do it sequentially, then everybody needs to do it that way, or does that matter at all?
I mean, from the study I did, it shouldn't matter. It's an interesting question.
Well, I'm just thinking, like, you want to eliminate as many variables as possible anytime you're you're working in this space. And so that could potentially be a variable, but it may not have an impact. I don't know. You're the expert for sure. I'm not, but
yeah. From the single study I did, it did make a difference.
Okay.
In a different situation, it possibly could. I don't know. But setting visual limits is a support function in a cleaning validation program. So most places don't have the luxury of resources to do that both ways, or to run that study, although it would be interesting.
Absolutely. So, Rich, how is determination of visual residue limits, there, I can say it out loud, how's that affected by the material of construction? Do optimal viewing conditions change with different materials of construction? So I can imagine that might impact it.
Absolutely. I mean, the obvious example would be PTFE or Teflon, which is a white surface. Yeah, you can't see much on that, no matter what you do. Stainless steel is a good surface for seeing visible residues, and that's typically what I do all of my work on. I did do a study with comparing stainless steel, Teflon, and glass, and yeah, Teflon you can't see anything, but glass and stainless steel had equivalent visual limits.
Okay.
The main difference between glass and stainless steel was that on stainless steel you can just lay it on a counter and look at it there. You try and bunch your coupons somewhat together so you get you want to try and mimic the equipment you're looking at, and the surroundings can have an impact on your ability to see the visual limit, particularly as you back away. So you worry about that, and that makes sense for glass, because if you have glass laying on a white countertop, well, it might as well be Teflon at that point.
Exactly.
Yeah, and a black background isn't much better. So for glass, the best way of looking at pieces of glass for cleanliness is just like you would do in your kitchen, or if you went to a restaurant, if somebody's looking at a glass, they'll kind of hold it up to the light and look at it from a fairly acute angle compared to the light to see if there's any spots on it, and that's what works best with visual inspections of glass pieces.
Yeah, that right. And I can imagine that, just holding it up. I do that with my own glasses some days. I'm like, what?
Yeah.
I know there's something there.
Exactly. And that work, and for small pieces of glass where that's easy to do, for larger pieces of glass where then you might not be able to pick it up, then you kind of have to adjust your viewing angle and try and work having that light source slightly offset in the background so you can get a better angle at it. So yeah, the best angle is different on a material of construction, and some it doesn't work. And the way to address that is using a cleanability study, and that's the ability to clean equipment, or you could look at your cleaning validation data. If you swab stainless steel and a light or white colored piece, and the best example of that is a gasket in a piece of equipment, you'll get swab data, and if your cleaning procedure cleans them to the same level, which I would expect,
right,
then if the stainless steel parts are visually clean, then you could assume that the other parts that you can't see as well are visually clean also. So you can tie that together. You have to use other things than just visual limits, though.
That makes sense. That makes sense, especially if you have Teflon components that it's very challenging to see any residues there. So, Rich, what steps should we take, or what steps need to be included for risk mitigation in VRL?
Yeah, you're going to measure closely the viewing parameters during your VRL determination, but when you're actually doing a visual inspection of a piece of equipment, you're not going to measure the distance or the angle. You could assess the light level on a periodic basis, I guess, but you have to train your people who do that to be aware of adequate light levels. Give them an idea what 200 lux looks like, and it's pretty dim. So that's pretty easy to assess that you have an adequate light level. You tell them to minimize distance as much as practical when they're viewing equipment. And most importantly, you want to tell them to look at surfaces from multiple angles as best they can. It's one example I had was when I was still at Merck, my counterpart in the commercial side said we had a failure here. The swab sample came out higher than the visual limit, and it's like, oh, that's interesting. And he actually had, the piece was small, the piece of equipment was small enough that I could borrow it and spike some residue on there and let it dry. And it was sitting on a bench top, and I looked at it, it's like, yeah, I can't see it, but I know it's there. Until I couldn't see it until I changed the angle I was looking at, and then it jumps out at you. So the angle is critical, and you want to emphasize that during your VRL training as best you can, because that is it's pretty astounding sometimes what you can and cannot see depending on the angle.
Absolutely. It sounds like it, especially when you know it's there and you still can't see it, right?
Right. Right. That's the trick.
Exactly. So with ever-changing technologies and regulations, including you just mentioned earlier that the agencies are starting to look for qualified people. Do you expect to see changes in the way VRL programs are utilized?
I would hope, but I'm not sure.
Well, maybe if you don't expect, what changes would you like to see to VRL programs?
Well, I think it's like you said, it's been 2 years since the agency started hitting people on visual qualification. So it takes a while for things to take hold in the industry. So companies will probably slowly come up with programs to qualify people to do visual inspections. You don't have to use VRLs. I'm not sure how else you would have some kind of quantitative measurement. But the agencies don't tell you how to do things. They just tell you have to do them, and then you have to be able to defend them. So I think visual limits are a good way to qualify personnel to do visual inspections. It's a great training tool. It's not something that takes a huge amount of resources. So it seems like to me the best way to go, but it'll just take some time to see how much it catches on.
Yeah, well, and I think it's important because this has to happen internally for each organization, right? Because
Correct.
It's not like you can go to a class to learn how to look for visual residue limits, because they're unique to each different manufacturing facility, each piece of equipment. So, one Merck might not have the same equipment that Pfizer has or whatever, right? So you have to train your people on A, the equipment they're using, and B, the actual products they're using.
That's correct.
So it's going to take some internal effort, I guess, is my point. It's not like a company can just come up with, some sort of program and we're going to send all our people to it.
Yes, you're right. Each situation you have to evaluate on a case-by-case basis, the combination of products and equipment, because and the facility itself. There'll be differences. A lot of it'll be the same, but there'll be differences. You're absolutely right, from place to place.
Well, Rich, as VRL programs are gaining importance again, I think it was great to have this conversation with you today and just learn a little bit more about how we can be proactive in terms of setting up our programs. Any last thoughts or ideas that you'd like to leave with our audience for today?
As far as visual limits, yeah, it's getting over, I guess. I don't know that most companies appreciate the contributions that VRLs can make to an overall cleaning program. Not only does it help to qualify your personnel to do visual inspections, but if you think about it, if you have, 30 products in your portfolio and a number of them are safe, if you validate the hardest to clean, you pretty much can just rely on visual inspection for your safer products rather than having to do swabbing or other testing or rinse sampling. So you can leverage visual limits to show that your equipment is adequately cleaned on an ongoing basis. So it almost like you're doing cleaning. You are doing cleaning verification every time you clean equipment, because you have to do a visual inspection. So the better you define that and qualify your people, the easier it is to defend that position that visual inspection is adequate for a given product for patient safety.
Very good. Great reminder for our audience. Well, Rich, thank you again for being our guest here today. It's been great to speak with you. I really appreciate you taking the time with us today. Thanks so much.
Thank you, Stacy.
Absolutely. I also want to give a shout out to our producer, Ben Kitchen. And lastly, a big thanks to you, our valued listeners. If you have topic requests or guest suggestions, we'd love to hear from you about that. And if you enjoyed this episode, we ask you to subscribe in your podcast player of choice and share with your friends, colleagues, and online networks. We really do appreciate you helping us spread the word. For today's show notes and information on this podcast, please visit www.ivtnetwork.com/podcast. We'll be back again next week with another great discussion. Until then, make it a great week.